So we've been looking at the place cell cognitive mapping system, and if you remember when we talked about the theoretical aspects of mapping, we saw that you need to have some type of compass information, and we've looked at the head direction cells. But you also need some kind of scale so that you know how distances in your representation relate to distances in the real world. So we know that place cells can compute distances, and there have been a number of experiments that show this, and you might remember this one by Gothard and colleagues, where they showed that place cells are able to compute the distance from fixed points in the environment. So in this experiment, one of the endpoints of that track was moved along the track, and we saw that the place fields moved along with it. So the place cell is able to compute how far the rat has walked since it touched the end of the box. And the question is, how do they do that? How do they know how far the rat has walked? So the answer to this came about more or less by accident. So in the process of trying to understand how place cells gather their information, people had been recording in regions of cortex that project to hippocampus, and we saw that that was how head direction cells were discovered. So in the sort of early 2000s, the Moser lab in Trondheim started exploring another region of cortex called entorhinal cortex. So we've seen entorhinal cortex before because it's the major cortical region that sends fibres into the hippocampus through the perforant path. And when we looked at LTP, we saw that the perforant path was a common target for stimulation. So it's a really massive input. And you can see it in green here in this diagram. And entorhinal cortex, not too much was known about it. So Marianne Fern, who's a PhD student, and Torkel Hafting, another PhD student, started recording from entorhinal cortex as animals were exploring a box. And in 2004, they published a paper showing that entorhinal neurons often show place-specific firing. It's a little bit like what place cells do, except that whereas a place cell will typically only have one firing field, one of these entorhinal cells would tend to have multiple firing fields. And you can see in the spike maps, the little red dots that reflect the action potentials, you can see that they're clustered in multiple regions. So they published these data and also presented them at the Society for Neuroscience meeting that year. And at that meeting, many scientists came and looked at these data and got talking. And one of them, Bill Skaggs, noticed that the spacing between those patches of firing seemed to be more regular than you would expect by chance. And he suggested that they try this experiment in a much larger environment. So they did this. And next year they published an absolutely groundbreaking paper showing that in a sufficiently large environment you can see that there's an extreme regularity to the organization of these firing fields. And you can see them in this picture here. That's showing 3 cells recorded in a 2-meter diameter, very large apparatus for a rat. And you can see that the firing fields are arranged in these very neat rows. In fact, what they do is they make a pattern that mathematicians call hexagonal close packed. So each one of those firing fields is surrounded by 6 identical ones. And they're very circular. So unlike a place field, which may be quite irregularly shaped, these firing fields are very circular and very evenly spaced. So they were named grid cells because they because the neat rows of firing fields suggested that this might be the function of them, to form a grid. And you can see from this schematic here that, as with place cells, a given grid cell produces action potentials in a given place in the environment every time the animal goes back there. So it'll go back there, a few action potentials will appear, then the animal will go away, and then it will come back and it'll lay down more in that place and so on. So to be able to accurately lay down its action potentials every time it returns to that place, it needs to be dynamically tracking the location and the path of the rat. And this, the result, as I mentioned, is this very regular hexagonal pattern. So are these a metric grid for the cognitive map? That was the kind of immediate assumption. It's turning out to be a little bit more complicated than that, and we'll look at that in the next lecture. Since the discovery of grid cells, many attempts were made to see how general this finding is, and so grid cells were recorded in different species. They were found in mice, almost exactly the same as they are in rats. Interestingly, the same size as they are in rats. So there aren't mouse-sized grid fields for mouse-sized animals. And similarly with baby rats don't have small grids either. So the grid seems to be a function of the physiology of the cells rather than being tied to the size of the animal. In human epilepsy patients who have indwelling electrodes to monitor their epilepsy, similarly to the experiments that we saw with place cells, when these people are recorded from while they're doing virtual reality navigation tasks, it's been found that in regions of entorhinal cortex, we see these grid-like, they're not as neat as in rats, but these sort of grid-like arrangements of blobs that, again, suggest that this is a general phenomenon, certainly across mammals, maybe broader than that. So this discovery really set seal on the idea that the hippocampal system is a mapping system, at heart. So it's not just a structure whose job is to associate stimuli in the environment. Space is really fundamental. And we know this because the grid cells tell us that distance, which is a purely spatial metric, is fundamental to this whole system. So in 2014, O'Keefe and the Moses shared the Nobel Prize in Physiology or Medicine for this discovery of a positioning system in the brain. So what we're going to be doing in the next lectures is looking at the properties of grid cells, and we'll see that they're more complex than we first expected, and that their function may be broader than we first thought as well.